Hello, I am Raghuvansh Prasad Sa, postdoc at University of Minnesota Medical School. The pathogenesis of pancreatitis is poorly understood. For over a century, we have known that trypsin activation is the central event. Early during pancreatitis, recently, endoplasmic reticulum stress has been found to be activated. What we do not understand is how endoplasmic reticulum stress, also an early event, and trypsin activation, another early event, how are they related? Trypsinogen activation takes place in special colocalization compartments where lysosomal fraction and zymogen granules colocalize, and the lysosomal enzyme cathepsin B activates trypsinogen to trypsin. In this study, we have used knockout mice which either lack trypsinogen or cathepsin B, and due to this genotype, these mice do not demonstrate pathologic trypsinogen activation. The objective of this slide is to demonstrate what are the molecular targets that can be studied in the lab which demonstrate ER stress. So to answer the first question, whether endoplasmic reticulum stress is independent of trypsinogen activation or it is actually a consequence of trypsinogen activation, we studied the markers of endoplasmic reticulum stress during acute pancreatitis, which was induced by cholecystokinin hyperstimulation for 10 hours, and compared between wild-type mice and trypsin knockout mice, trypsinogen 7 knockout mice, which do not have trypsinogen activation. So as compared to controls, you can see all the markers of ER stress demonstrate that ER stress is activated in acute pancreatitis in the wild-type mice in the presence of trypsin activation, and also in trypsin knockout mice which do not have trypsin activation. They have equal demonstration of equal evidence of ER stress activation. For chronic pancreatitis, the scheme was to induce acute pancreatitis 2 times every week for 10 weeks. We found that endoplasmic reticulum stress is persistently present in chronic pancreatitis. As you can see, marker of ER stress, GRP78, also known as BIP, was expressed at much higher levels in chronic pancreatitis compared to controls, and this was also demonstrated by immunohistochemistry. Other markers of endoplasmic reticulum stress, phospho-PRK, phospho-EIF2 alpha, ATF4, are all upregulated and persistently present. Now the objective was to see if this persistence of ER stress in chronic pancreatitis is also independent of trypsinogen activation. So we used the cathepsin B knockout mice and trypsin knockout mice, which do not have pathologic trypsinogen activation, and compared chronic pancreatitis and ER stress markers among this group and the wild-type group versus the saline-injected controls. So what you can see here compared to controls, GRP78 CHOP, both markers of ER stress, are upregulated in chronic pancreatitis, and same degree of upregulation can be seen in wild-type, cathepsin B knockout, and trypsin knockout mice, suggesting that ER stress persistence in chronic pancreatitis is also independent of trypsinogen activation. So we have shown that endoplasmic reticulum stress is activated in acute pancreatitis as early as trypsinogen activation, and it is actually independent of trypsinogen activation. We have for the first time also shown that endoplasmic reticulum stress is persistently present in chronic pancreatitis, and this is also independent of trypsinogen activation. Thank you.